I believe in science because it's what provides answers. It's what have created the medicine that have enabled us to live the life that we live today. I think research is absolutely essential for us to thrive in our environment, to do what we like to do on a daily basis. I am Victor Torres, and I'm a microbiologist. In my laboratory, I study the bacterium Staphylococcus aureus, also known as MRSA or MRSA. And the reason why we study this organism is that over the years, it has become into an antimicrobial resistant organism. The fight against multi-drug resistant pathogens is becoming very serious. The emergence of these microbes is increasing while the development and rollout of the drugs is decreasing. So this is a fight where the bacteria are winning in masses. One of the strategies that Staph aureus uses to cause infection is that it produces a number of molecules, particular toxins called leukocidins. So these leukocidins are toxins that selectively kill the immune cells that we have patrolling our body to protect us from infection. So leukocidins are fascinating because what they do is that they recognize the plasma membrane of the cell that they're going to attack. They bind to the surface of the cell and then they assemble into a little pore, a little hole. Imagine having a balloon and taking a pin and pinching the balloon with the pin, the balloon will explode. Some Staph aureus strains that infect us can produce 5 different leukocidins. And one of the discoveries of the lab was that we identified that each toxin has kind of like a specific zip code to target different cells. So they don't target all the cells in our body. They're fairly specific to the subset of cells that they can target and eliminate. If we prevent the toxins from working, if we can block them, can we boost the ability of the host, those immune cells, to kill the pathogen? We generated small peptides called centurins that bind to the toxin, block the toxin from recognizing the plasma membrane of the immune cells, and as such protected the immune cell from the lytic activity of the toxin. The question then becomes, does the host do something to protect itself from these toxins? And by studying that interaction, what we discover is that indeed, when a pathogen infects a host, us, our cells respond by releasing exosomes. Imagine flares that are released to distract the toxin, to distract the pathogen. In this case, they're like sponges that will suck up the toxin, preventing the toxin from interacting with the receptor and killing the cell. So what this project tells us is that the leukocidins are indeed a valuable target. We can target them with small molecules. Most likely we can target them also with vaccines to try to contain Staph aureus infection. What is unique about the research that we do is that we're not constrained by a discipline. We really deploy everything that is available in 2021 to try to tackle the questions that we're interested in. Our research program has one goal and one goal only, and that is the hope that something that we discover will at some point help the life of people.